talk about what those things are today and why they matter, why we even care. And they're things you may not know, remember the terms. Electron transport, you probably do remember this term from various biology classes. You may not remember oxidative phosphorylation, but they're important terms that as soon as we talk about them, you'll be like, oh yeah, I remember that. Anyway, I was going to tell you guys some other news. Does anyone, so I read fantasy books, I love fantasy literature, I love fiction, science fiction. Does anyone read Brandon Sanderson books? Anyone? No? Dang, there go your bonus points. All right.
Which one?
Before we get into glycolysis, there's something we need to finish. Something we need to review. Okay. There it is. And Cyril, you asked for charge stabilization, right?
Yes, sir.
It's a good question. It's a good question because this same concept of charge stabilization is going to come into play in today, maybe one, in at least one of the steps, pathways of glycolysis, and at least one of the enzymatic reactions that we're going to talk about today. Probably two if we get to step 3 today. This idea of charge stabilization, while not the exact same thing, the same exact effect is going to play a big role in these two enzymatic pathways. We'll talk about one for sure. We make it. So, talk about 4 different things, reasons, explanations for the large negative delta G value in the hydrolysis reaction, phosphoryl transfer of ATP of a phosphoryl group from ATP. The second one we talked about was charge stabilization. And what does this mean, charge stabilization? You have charges want to be stabilized, right? Charges want to be stabilized, and if you have a positive charge, it wants to be stabilized by negative charges. And this partially positive charge is a pretty big partial charge, right? Why? It's surrounded by a lot of oxygens, right? A lot of oxygens. So if you look at the oxidation state of that phosphorus, it's probably going to be pretty positive. In fact, it's surrounded by a lot of negative, electronegative oxygens. So if we have, if we look here, this partially positive charge is stabilized in this case by one negative species. This partially positive charge is stabilized directly by one negative species. However, once this is hydrolyzed, you can see this partially positive charge is stabilized by 2 species. This partially positive charge is stabilized by 2 species. Does that make sense? It just gives a bigger shield around it, a bigger buffer around that partially positive charge. And it does affect the chemistry. It does affect the chemistry around this. We'll talk about that in a minute. We will talk about that in a minute. Does that help?
Yes, sir.
Okay. Last thing we need to finish is our little talk about metabolism. I told you I'm not going to go over various terms of metabolism or of different modes of metabolism, different types of organisms. These are things you've probably talked about at this point, at least in 2 classes, probably 3, maybe even 4 classes, depending on what you've taken. Those things aren't. It is necessary to understand the entire concept of what's going on in this big picture. It's not necessary to understand step 1, step 2, step 3, step 4, step 5, all the way through step 10 of glycolysis, but to understand where glycolysis fits into the large picture of metabolism and energy production in a specific organism. These things are necessary to know. We talked about anabolism, catabolism, 2 different directions of metabolism, right? 2 different directions. One, anabolism, the synthesis of biomolecules, of big things from small things. Metabolism, the degradation of big things into small things. One of these pathways is divergent. You start with a few building blocks, and you can build many different things. The other one is convergent. You start with all these many different things, and you can break them down into building blocks. One of the things we didn't really talk about is if both of these are going on simultaneously all over the place, you can see that they would kind of be competing for resources at some point, don't you think? One thing is pulling one way and the other thing is pulling the other way. Something's going to give at some point. You see a lot of these pathways are compartmentalized in different locations. So a lot of these processes are occurring in the cytosol. A lot of catabolic processes occur in the cytosol. A lot of anabolic processes occur in places that aren't necessarily floating around in the cytosol of the cell. They're occurring in other locations. Why? So these processes are clashing with each other. Based on what the cell needs at that time. The steps of catabolism. So the pathways we're going to talk about are catabolic pathways for the most part. Anyway, for the most part, we're going to talk about catabolic pathways. Three. What's up?
The divergent and convergent.
Mm-hmm.
You switch it.
No, it isn't. No, that's right. Anabolic pathways are divergent. So anabolic pathways, you're building materials, right? You're starting with a few starting points.
Yeah.
A few starting points. So anabolic pathways are definitely divergent. You start with a few materials, and you build many different types of things, right? So you have 3 or 4 starting points, starting base materials, and you can build how many different proteins, how many different enzymes, how many different polysaccharides, how many different lipids. You can build a lot of different things. No, absolutely, that's right.
But what of catabolism and convergent? I thought this convergent is like...
It is. All these catabolic processes are breaking down big things, right?
Yeah.
Into products.
Into
the
many
same 3 products. The same 3 products. So you have all of these different things converging into water, converting into ammonia, converting into carbon dioxide. Everything is made up of water, carbon dioxide, and ammonia.
Okay.
Those are your 3 sources. So yes, that is right. I think I said it wrong the first time, but that is right.
Okay.
Catabolic pathways are convergent. They are breaking down many different big things into the same starting materials. Does that make sense?
Yeah, I thought of it different, like catabolism, since it breaks down into many products, like you have a single compound break down into separate.
It does. But you have, think about how many different types of enzymes you can build, and what do you use to build those enzymes? The same 20 amino acids. So you can literally break down 500,000 different types of enzymes into the same 20 building blocks. And you can take those building blocks and break them down into the same 3 starting materials. So literally, out of 3 things, you can build 500,000, about what we've counted so far, different proteins. That's just proteins. Out of 3 things, you can build hundreds of thousands of different things. Does that make sense?
Yeah.
So when they're breaking it, and hopefully this will help. So stage 1, big things. Nutrient biomolecules. Big things, there are many of these. Things like protein, polysaccharides, lipids. So nutrient biomolecules, big things are broken down. Building blocks. Polysaccharides, for example, big things are broken down into polysaccharides are made up of many different monosaccharides, right?
Mm-hmm.
So polysaccharides are made up of monosaccharides. So you have many things being broken down into one thing, basically, right? So if you have a polysaccharide made up of many different single monosaccharides, right?
Mm-hmm.
So you have big things broken up into smaller single pieces. Stage 2, these building blocks right here. Are broken down into 2 things. Literally, this is where it all converges into 2 things. These building blocks are broken into pyruvate and acetyl-CoA. We're going to get to pyruvate today. We'll get to acetyl-CoA probably sometime next week, Wednesday, maybe Monday next week. So these building blocks, let's take polysaccharides. Broken down into monosaccharides like glucose. We're going to talk about glycolysis today, right? The breakdown of glucose. Glucose is further broken down into pyruvate. Pyruvate eventually gets to acetyl-CoA and goes to the TCA cycle. Once we get to small things like pyruvate, acetyl-CoA, these are converted into ATP, water, CO2, ammonia. Why do we call this a convergent pathway? Because literally all of the things you start with, all of the different proteins, all of your different polysaccharides, all your different lipids are broken down into essentially 4 things: ATP, energy production, electrons, which will assist in energy production, water, carbon dioxide as well. The first step, the first pathway that we're going to talk about is glycolysis. Glycolysis is the degradation of glucose. The degradation of glucose leaves us with some products. The products we're left with are, these are important products. We're left with 2 pyruvates. So glycolysis breaks down glucose, leaves us with 2 pyruvates, a net gain of 2 ATP. 2 ATPs and 2 NADH. It is a 10-step pathway, and we're going to learn all 10 steps. We're going to learn the enzymes associated with each step. We're going to learn what type of reactions are taking place and why they're taking place at each step. It's a 10-step pathway, and we're going to break this down step by step by step. And it's really divided into 2 phases. One is a priming phase. The other textbook, I think the literature textbook calls it a preparatory phase. Either way, you can think of it. One is a phase that doesn't produce anything; it actually requires material. The example in your textbook is, imagine priming an old water pump. If you want to get water out, you have to do some work. Put some work. Eventually, something comes out. You got to do something. So you do some work in the priming phase. It requires ATP to get to a certain point, and then we are left with a payoff phase, in which we get back more than we put in. The priming phase requires 2 ATP, minus 2 ATP. So the priming phase requires 2 ATP. The payoff phase generates 4 ATP. You can imagine that's a net gain of 2 ATP, right? It generates 2 NADH. It generates 2. The priming phase are steps 1 through 5. Payoff phase are steps 6 through 10. We're going to go through all 10 of these steps. Enzymes required, why these are happening, how they're happening, inhibitors, regulation steps, how this entire pathway is controlled. And then we'll talk about what are the fates. So after this chapter, we'll talk about what are the fates of pyruvate, what are the fates of NADH. Those are the 2 other things. So this pathway, we're talking about how do we get to 2 pyruvates, how do we get to 2 NADHs, how do we get to 2 ATPs, net gain. And the next pathway, the TCA cycle, again, these are cycles we've talked about, right? We've talked about these cycles. Brushed over these next steps. What's involved? What outcome? What is the end product of these reactions? Pyruvate is destined for TCA cycle, Krebs cycle. Under what kind of conditions? Anaerobic or aerobic?
Aerobic.
Aerobic, right? So that's the aerobic pathway. So what are the fates of pyruvate? Depends on availability of oxygen. Glucose, which is C6. Glucose is broken down into pyruvate. Right? 2-pyruvate. Be able to draw pyruvate. You should be able to recognize pyruvate. Broken down into 2 pyruvates. In this process, we are generating 2 other things, right? We generated 2 ATPs. 2 NADHs. What do we call this? This is glycolysis, right? And this is a 10-step process that we're going to, as soon as we're done with this page, we're going to get into these steps, the 10 steps of this process, the 10 steps of this pathway. But what happens to pyruvate? What happens to pyruvate? Under aerobic conditions, under aerobic conditions, this reacts to form an additional 2 NADH. So an additional 2 NADH to form 2 carbon dioxide and 2 acetyl-CoAs. This will go through the TCA cycle. We'll talk about that next chapter. Under certain anaerobic conditions, certain organisms, under anaerobic conditions, organisms, microbes, large animals, humans, under poor oxygen conditions, will convert pyruvate and NADH to NAD plus. So the 2 NADs that we just produced are used in an anaerobic process to convert pyruvate to 2 nitrates. Other organisms like yeast, this anaerobic process leads to the lactic acid cycle, right? Which regenerates NAD to be used again in glycolysis. Things like yeast undergo fermentation, alcoholic fermentation, in which the same process, 2 NADs, 2 NADHs, are used to convert this to CO2 and 2 ethanols. So what is different about these 3 pathways? What is different about the aerobic pathway from the two anaerobic pathways? Obviously the presence of oxygen, right? That was in the definition.
In the aerobic pathway, we're creating more NADH.
So here's the key. Here's the key. In the aerobic pathway, we're actually creating more NADH. In the both of the anaerobic pathways, we're taking what NADH we just produced and chewing it back up and creating NAD plus, right? And there's a reason. There's a reason, and it's a useful reason. But we need to compare oxidation of existing NADH versus reduction of additional NAD. In one pathway, we reduce more NAD plus, right? In one pathway, we reduce more NAD plus, and in the other two pathways, we're actually oxidizing the existing NADH that we just made. And we're going to talk about later. I told you we'll talk about destination of pyruvate, which we're going to, right? TCA cycle under aerobic conditions. We'll talk about destination of pyruvate. We're not going to go back into the lactic acid cycle. You've seen this in other classes, and that's not something we're going to do in this class. We're going to talk about the aerobic pathway from here on out. But we'll also end up talking about what is the destination of these NADHs and what do we get from these NADHs. Including the entire aerobic pathway, you can get anywhere from 30 to 38 ATPs from one glucose. From one glucose in the aerobic pathway, you get about 38, anywhere from 30 to 38 ATPs. In an anaerobic pathway, you get two. Anaerobic pathway, you get 2 ATPs. Aerobic pathway, you get 30 ATPs. So why? That begs the question. Why in the world? I mean, if that's what you're comparing, even under anaerobic conditions, my goodness, just keep doing this, right? Keep doing this until you run out of oxygen. That's what you would think, right? However, even if you're just settling for 2 ATPs in the aerobic pathway, without the presence of oxygen, you are going to, and hopefully this will make sense by the time we get finished with all these pathways. By chapter 20, hopefully this makes sense. In the aerobic pathway, what would happen if we ran out of oxygen? If we ran out of oxygen in the aerobic pathway, we are eventually going to run out of NAD plus. NAD plus. And what I didn't draw here, and I probably should have, first step, well, not the first step, one of the critical steps of glycolysis requires NAD plus to produce NADH. If we run out of NAD plus, we stop. Glycolysis stops, and we stop our very first step of metabolism of glucose. If this is obligate anaerobes can only, or obligate aerobes can only follow one pathway or the other, right? Facultative anaerobes, like us, both can operate and can follow both methods. So under poor O2 conditions, we can actually follow another method, right? Skeletal muscle tissue under stress can follow either method. In a worst case scenario, this pathway can end up regenerating NAD plus. So even if this is as far as you get, you can go back and produce two over and over and over again. So the pathway under worst case scenario doesn't stop. You can always regenerate NAD plus. Does that make sense? Hopefully it'll make more. If it doesn't make sense, doesn't make a lot of sense. Hopefully it'll make more sense by the time we finish electron transport chain. All right, let's get into our priming phase of glycolysis. I told you this is going to take 2 to 3 days to go through, and it probably will take 2 to 3 days. My guess, we've already spent 30 minutes, so we have 20 left. We're probably going to get through 2 phases today. What's up?
We like stop you before you get ahead because you only got 4.
Yeah, pull out a different page around on the back. I'll post the notes today. If you want to print them off. If not, literally, this is what the notes from here on out look like. Phase 1, reaction 2. Reaction 3, hey, this is a whole page. It took me a lot, I spent a lot of time typing up these notes on this page. Reaction.
Took you all night up in the kids.
Yeah, I was up late. Reaction 4, this took me twice the amount of time because I typed that and that. No, I didn't. That does that automatically whenever I start a new page, so I did not do that. This actually didn't take me any time because I just copy pasted it from phase 1 and changed phase 1 to phase 2. I joke. Okay. Phase 1. Priming phase. Phase 1 is not going to produce energy, right? It's not going to produce energy, and it is going to require energy. See, right? Priming phase, preparatory phase, however you want to think about it. It's not going to produce energy, it is going to require energy. And step 1 is the phosphorylation of glucose. Step 1 is a phosphorylation of glucose, and we're not going to draw this mechanism. Why are we not going to draw this mechanism?
We already did.
We already did it, right? When did we do it? What did we title this mechanism?
It's hydrolysis.
What? Phosphoryl transfer. But when we did it, it wasn't under a section called phosphoryl transfer. Hydrolysis of ATP, right? Hydrolysis of ATP. When we did the when we drew this mechanism for the hydrolysis of ATP, I told you, and I said, imagine instead of H2O, this is an R group and then an OH. Guess what? Glucose has an R group and an OH, right? It has a primary alcohol in the end. It has a primary alcohol. It is the same reaction mechanism. The primary alcohol on glucose follows that exact same mechanism that we drew, so we're not going to draw it. Step 1 is, let's see, this is the ding in the priming phase. It's not really a ding, it's not at every step, it is at a couple steps. This step is thermodynamically unfavorable. This isn't new, right? We talked about this when we talked about bioenergetics. Many of these biological processes are thermodynamically unfavorable. What are a couple ways we can overcome this energy barrier? Removing product or
Coupling.
coupled reaction with something that is energetically favorable like ATP. All right, so this reaction is coupled with ATP hydrolysis. Right? So the phosphorylation of glucose is going to be coupled with ATP, so we'll get phosphoryl transfer from the ATP. You did this one, right? Delta G naught for the phosphorylation of glucose. We call positive 13 point something. I think it was 13.8. Delta G naught for the hydrolysis of ATP, something like negative 30.5 kilojoules per mole. Gave us a overall delta G naught. I'm trying to do math here. 16.7 kilojoules per mole. But the unique thing is that the actual delta G is even more favorable. Negative 33.9 kilojoules per mole. This first step, this first coupled reaction is very favorable biologically. Why? Why do we want to phosphorylate glucose? One of the main reasons we want to phosphorylate glucose, phosphorylated glucose is membrane bound. It's membrane bound. Glucose can diffuse through a lipid bilayer. We haven't talked about membranes, which so some universities offer like a Biochem Light type class where who's taking Biochem 2? Who's going to? A lot of you guys are going to take Biochem 2. You're taking Biochem 2? You're taking Biochem 2? Okay. A lot of people are going to take Biochem 2. That's surprising. In the other class, everybody's like, no, we're just taking Biochem. What?
It's all that's required as a prereq for med school.
For med school, right. So most people only think about them like a lot of universities only offer like a Biochem class. And in that class, they go over things. They don't like. They do a lot of like hand waving. This happens without explaining. It's. They talk more about what's useful to med school, and it would kind of be neat. In this case, it would be something that would help. If you talked about membranes before, which we haven't talked about membranes, we'll talk about membranes and biochem too. More needed here. Glucose can diffuse through a membrane. An uncharged glucose molecule can freely diffuse through a membrane. However, once you stick a nice charged phosphate group on the end of it, phosphoryl group on the end of it, it cannot diffuse through a membrane. So what does that do? It actually makes this a nice place to regulate because it's not going to stop at some equilibrium. Glucose coming into a cell and diffusing through a cell is never going to find equilibrium. Why? Because glucose diffuses into a cell, and guess what happens? It changes. So guess what's going to happen to more glucose? It's going to constantly try to come into the cell to reach that equilibrium. Why? Because you're drawing it away. As soon as it comes in, it's being drawn away to something else, and it's stuck in a cell, right? You can't leave. So there is no, you kind of get a sink pulling glucose in, a thermodynamic sink, if that makes sense. When I say sink, I'm drawing something towards someone else, not something that you do dishes and sink, pulling something in, it's pulling glucose in. This product, so what is a product? A product is glucose is going to glucose 6-phosphate. This reaches a branch point in glucose metabolism. Glucose 6-phosphate represents a branch point in metabolism. It can follow catabolic pathways. It can follow anabolic pathways. So glucose 6-phosphate basically has a choice at this point. Follow glycolysis, which we're fixing to talk about. Pyruvate. Follow pentose phosphate pathway, which we'll talk about later. And lastly, follow an anabolic pathway, leading to the formation of complex carbohydrates and energy storage. And we'll get to that in a second. Pentose phosphate pathway. Don't worry about it yet. We'll get to it later. Pentose phosphate pathway. That's something we're going to talk about. Not yet. Two enzymes catalyze this reaction. Actually, one enzyme catalyzes this reaction. That's a big difference, right? One enzyme catalyzes this reaction. This enzyme has various isoforms, right? We call those isozymes. What other enzyme is an isozyme that we talked about? Remember? Yeah, that's I'm impressed. I'm impressed you knew that. Hopefully you did. It's important. Lactate dehydrogenase. Remember, I told you is like a poster child for isozymes. If there was a poster child, it would be LDH. For lysozymes, or isozymes, I'm sorry. LDH is unique because it can actually catalyze the same reaction in different directions, right? One isozyme catalyzes lactate pyruvate, where the other one catalyzes pyruvate to lactate, which is very, very, very unique. Not only are these isozymes similar, they do the opposite. It's not that they do different chemistry, it's that they do the exact opposite. Kind of neat. Various forms of of kinases, of glucose kinases are found in different parts of the body. Hexokinases are found in pretty much everywhere. Glucokinases are found predominantly in the liver. Hexokinase. So again, these are both isoforms of hexokinase. So I think hexokinase found in skeletal muscle tissue is a combination of type 1, type 2, and type 3 some combination of those three, whereas glucokinase is the type 4 hexokinase, and they're found predominantly in the liver and the pancreas. What is the difference between these two? The main difference between these two is hexokinase. You can imagine one is called hexokinase, one is called glucokinase. Glucokinase is more specific to glucose. Hexokinase is specific to hexoses, right? So pretty much any six-membered sugar, pretty much any six-membered monosaccharide. Hexokinase is not really specific to glucose. It will bind these with a Km 0.005. I'm sorry, I have too many zeros there. 0.05 millimolar. You may see different values reported in your textbook. Just assume that I'm always right. The Km for glucokinase is around 5 millimolar. Is that a big difference? It is a massive difference, right? That is 2 orders of magnitude difference. That is a large difference in Km values. A very, very large difference in Km values. So what does it matter? Why do we care? It matters. Why does it matter? We're going to plot activity of our kinase. What do kinases do? Phosphorylate, right? So if you miss a question asking about a phosphorylation step, and you circle an answer that's not a kinase, I'm going to get mad. So if I ask what enzyme represents step 1, the phosphorylation of glucose, and you have 5 different answer choices, and you circle one that's not a kinase. If you get the wrong kinase, that's fine. But if you don't circle the kinase, I'm going to get mad. Why? Because that's what kinases do. At this point, you should know that. There's one oxidation step and I guarantee you'll see this on your exam. Which step in glycolysis, or what enzyme in glycolysis, what of the enzymes in glycolysis represents the step number 6, the only oxidation step in glycolysis. If you don't answer dehydrogenase, I'm going to be mad. There's one dehydrogenase, right? Dehydrogenase do oxidate. We already talked about those are oxidoreductases, right? They do redox chemistry. There's only one. So if you circle a kinase, I'm going to get mad because that's not what kinases do. So if we plot activity versus glucose concentration, which one is going to reach saturation faster? Hexokinase. Is that the better Km? If we were looking at kinetic parameters, which one of these has the best kinetic parameter? Hexokinase, right? So that was one of your questions on your exam. Just like we said, many of these enzymes do chemistry on various substrates, right? We call whatever they do best at their native substrate. So hexokinase does this chemistry with different substrates. Which one does it do best? How would you measure those? You can measure those as a function of Kcat and Km, right? V over K is our efficiency number, right? It's an efficiency value for how well does this do. Compare Km, compare velocity, or Kcat. So if you're comparing both of those things, it's a good measure of which one is your native substrate. So low Km values are better, right? Low Km values are better. High Kcat values, high velocities are better. That was one of your exam questions. I think a third of the class missed that question. It was just understand what steady state kinetic parameters mean. So low Km values are better Km values. Hexokinase is going to reach saturation faster. So if you plotted this, you would see something like hexokinase reaches saturation really, really fast, whereas glucokinase is not. Km for my hexokinase would be way down here. Km for my glucokinase would be way here. Km for glucokinase, Km for hexokinase. And if I told you that resting, fasting blood sugar levels were probably somewhere around here. So this is, should probably label what that is, not just scribbles. Fasting blood glucose levels. Which one of these enzymes responds well to eating?
What?
The bottom one, right? If let's say after a meal, obviously this value is going to change, isn't it? After a meal, this value is going to change. Glucose concentration is going to shift up. Does hexokinase do anything differently? No, it doesn't, right? So hexokinase is not a good response to metabolism of glucose during high concentrations or post-meal. A lot of this is taken care of by glucokinase. Why? Because activity of glucokinase is very, very, very dependent on concentration. A few other things. Very important about this. Oh my goodness, I was thinking we were going to face step 3. We're only going to do step 1. All right, we'll finish with step 1. Let's get done with this. Hexokinase. Features product inhibition. And product inhibition is not a, it's not exactly rare. I wouldn't call it common, but it's not uncommon. It may be uncommon, but it's not rare. If you play video games like World of Warcraft, you would know the difference between those things, right? That was a stupid joke. Speaking of World of Warcraft, BlizzCon announced that they're going to open up legacy servers next year. Nobody. So you don't read and you don't play World of Warcraft. You guys, no bonus points again. Sorry for you guys.
If I played World of Warcraft, I would.
What's that?
I just had Overwatch.
Oh, nice. Overwatch is getting respect. That's good.
There's a really good South Park episode on
There's a
World
really
of
good.
Warcraft.
Oh, there it is. It is fun.
That's true.
Yes. You know how many? So that came out when I was in college, like my last year of college. You know how many people messaged me? How many of my high school friends that knew I was a nerd and played video games? Like, did you see me in South Park? Yes, I did. That's not me. My mom no longer cleans up my poop. Bathroom. No. Hexokinase features product inhibition. It's not rare. It may be a little uncommon, but it's not rare. So what does product inhibition look like? In the concentration of glucose, you can imagine if the concentration of glucose gets higher and higher and higher, glucokinase kicks in, starts working more. The concentration of glucose 6-phosphate is going to go up, right? If these enzymes start doing more, the concentration of glucose 6-phosphate is going up. The product of hexokinase, that product will allosterically bind to hexokinase. And will allosterically bind to hexokinase. So what do you see in product inhibition? You actually see at high concentrations of substrate, which would be high concentration. You can assume that's going to correlate to high concentrations of product. When glucokinase kicks in, at high concentrations of product, this will actually bind allosterically and start to inhibit. This is our first regulation step. Of glycolysis. So this is our first regulation step of glycolysis. We're going to talk about 3 regulation steps of glycolysis. This is our first regulation step of glycolysis. Product inhibition, hexokinase. Glucokinase. It's only relevant at high levels, right? Why? Because it's PM is so high. It's only relevant at high levels of glucose. This is going to yield when glucose kinase kicks in, high levels of glucose. This is going to yield an abundance of glucose 6-phosphate, right? Which that's going to turn off hexokinase, right? The abundance of glucose 6-phosphate is going to turn off hexokinase, but what are they going to do with this abundance of glucose 6-phosphate? This is where another pathway kicks in right here, right? Glucose 6-phosphate in abundance is going to follow an anabolic pathway. It's going to be stored as energy as glycogen in the liver. And the last thing we're going to talk about The last thing is we're going to talk about, and the only reason I'm going to push through and we're going to talk about this is because I told you we were going to talk about this. Talk about this charge stabilization shielding effect. Hexokinase does not bind to ATP. Hexokinase facilitates glucose. So I drew this right here. That's totally right. Glucose plus Mg ATP. The glucose 6-phosphate plus ATP. So not glucose. Glucose. Oh, wow. So hexokinase is actually specific to the magnesium ATP complex, not just ATP. It's actually specific to the magnesium ATP complex. And why? Why does this help? Two things. One, it's specific to the magnesium ATP complex, Mg2+. So this magnesium complex, right, it stabilizes ATP. And this magnesium complex, it shields the negative oxygen charges, right? So these negative oxygen charges, we said a second ago, these negative oxygen shield our partially positive phosphate, right? These negative oxygen charges, what's up?
What's specific to the magnesium complex?
Hexokinase. And glucokinase, kinases, hexokinases. So both of these are hexokinases, right? These hexokinases are specific to this complex. But why this complex? Why not ATP? So this magnesium charge. So we said a second ago that these negative oxygen charges help stabilize this partially positive phosphorus, right? It helps stabilize this partially positive phosphorus. So these negative oxygen charges make this phosphorus a worse electrophile, right? It makes it a worse electrophile. We said the first step in hydrolysis is that there phosphoryl transfers a nucleophilic attack by an OH group onto an electrophilic phosphorus. And these oxygens do not help that. They do not help that process. So what would help that? If we can draw that charge away from that phosphorus, and that's exactly what these magnesiums do. So magnesium charges shield these oxygens on ATP from a partially positive phosphorus, right? From a partially positive phosphorus, making it a better electrophile. When I say it, I mean that partially positive phosphorus, making it a better electrophile. The last thing I'll say about this is, and it is kind of important, so I can make a little bit. Hexokinase follows an induced fit model, and of course, we'll say they all follow an induced fit. Upon binding ATP, you see a massive conformational change. Why? Massive conformational change. This conformational change basically protects ATP from water. So this ATP cannot be hydrolyzed by water to produce inorganic phosphate. It facilitates the transfer of ATP to glucose and not ATP to a water, just to release inorganic phosphate. It actually, this induced fit, what is the purpose of this induced fit? It's to shield the ATP from any water molecule. Where is this happening? Cytosol of the cell, right? There's water. There's water available. So the purpose of this enzyme, the purpose of the conformational change is to protect this ATP once it's found from water. All right, see you guys on Friday.